I would like to introduce today's speaker. Dr. Florent Trouillas is Cooperative Extension Specialist with the Department of Plant Pathology at UC Davis and the Kearney Agricultural Research and Extension Center. Today, he will be speaking on the epidemiology and control of the invasive red leaf blotch disease of almond. Florent, you can go ahead and share your slides now.
All right. Well, thank you, Peter, for this opportunity. So yes, I'm an extension specialist with UC Davis. I'm a plant pathologist. I work with fruit and nut crops. I'm typically the kind of guy you would call when there is something unknown in the orchards where trees are dying and you want to try to have an answer. And that's how red leaf blotch came my way. Red leaf blotch is new to California. It's 2024 was the first detection. And so today the goal of my talk is to provide a general overview or current knowledge on red leaf blotch. We have conducted about one growing season of field research, so we have a lot of research updates, and then we have also have common knowledge, general knowledge coming from Europe. So the goal of my talk today is to present sort of a summary of the latest knowledge we have on red leaf blotch for California. And again, it's a perfect timing for me to give that webinar because disease management for red leaf blotch is coming into consideration within the next few days or weeks. So perfect timing for me to go over red leaf blotch right now. So red leaf blotch is an old disease of almond. It's a disease that has affected almond trees throughout the Mediterranean regions. It's very important disease of almond in Spain, region of the Middle East. The first detection, however, was from France, 1843. And again, it's a disease that only affects the leaves of almond, all right? It's not getting on the twig, it's not getting on the hull, on the fruit, really only a disease of leaf. It's caused by a fungal pathogen. The pathogen is called Podistigma amygdalinum. And again, for California, I got a call basically around May 29, 24 from Merced County, a farm advisor, Cameron Zuber, who basically sent a picture just like this one asking, hey, any idea of what's going on here? And so, yeah, quickly enough, we realized we had this red leaf blotch making its way in California, and again, really acting as something new and invasive for the state. So here I like to share some of the early symptoms. That's a disease that's going to show up in the orchard around the last week of April, first week of May. What you see here on these pictures are just a few yellow spots at the onset of symptom development. And then you see also that these sort of yellow spots can come together to sort of coalesce and form these orange color lesions. These are sort of the similar pictures in the field. And what you see now, hold on, let me pull out the pointer. What you see now, I like to show this sort of example here. This is a cluster of leaf with many yellow spots, and all these various spots basically indicate single spore infection from the pathogen Polystigma, right? So you can see really the high disease pressure within that orchard here. We're in Merced County with every little spot basically, yeah, again, illustrating one single spores of the pathogen landing. So, it seems like, already within a few years of introduction of this disease, we already have really high disease pressure in some of our Californian almond orchard for Polystigma. I should have said earlier also, it's a disease of almond only. I often get the question, will it affect the peaches? Will it affect any other stunt fruit? No, red leaf blotch is a disease really specific to almonds. I always like also to illustrate symptoms for field diagnostic. I would say red leaf blotch is pretty unique, but there's been a confusion at the onset of symptom development What you see here are the typical symptom yellow spots of red leaf blotch. And here on that same leaf, you see rust. Rust has been common this last few years also in almonds. And so you see here again more spots, yellow spots of rust. But rust can be easily diagnosed in the field if you just turn the leaf around. If you look at the underside of the leaf, you will find the typical sporulation of the rust, right? So again, here down here, you have a red leaf blotch, sort of more diffuse yellow spot and sort of sharper, tiny yellow spots of the rust. So something easy to distinguish between RLB and rust. And here what we see here now are sort of some yellow spots with usually a necrotic center. And those are the symptoms of Pseudomonas from the bacterial blast pathogen. Sometimes the bacterial blast pathogen will just not only affect the flowers, but it will also spread onto leaves. And so when Pseudomonas affect the leaves, this is what you see, a sort of a yellow halo surrounding sort of a necrotic central lesion. Now we're in the middle of the growing season. We're now in June and July, and that's how red leaf blotch looks at this time of the year. Bright orange color, multiple lesions on the leaves. If you see generally a lot of leaves may be affected within an orchard. And as we move a little later towards the middle of the summer, then these lesions start to become necrotic. They're sort of grayish and brown, and eventually the leaf will sort of curl up and eventually drop. So this is for the sort of midsummer symptoms on leaves. Remember those bright orange color lesions. Once you've seen it, you know red leaf blotch is quite a very easy disease to diagnose in the field. And so why red leaf blotch is a concern to our almond industry is that the pathogen is really aggressive to our almond cultivar, right? This is a block in its most likely second year of infection. The block was not sprayed, not treated with any fungicide at the proper timing. And this is what we see then. Pretty much just every leaves on every tree in the block was affected by red leaf blotch. And, like this advanced symptom, it looked like those leaves were almost burned, right? So you can determine just by this picture how aggressive red leaf blotch might be to our almond cultivar here in California. Now we're moving a little later in the season. What some of the highest risk with red leaf blotch is early defoliation. So we're now towards August, getting close to harvest, and at this point, some of these almond trees have lost all their leaves. Okay, and so early defoliation, of course, is a concern as the tree may try to push some new growth. And pushing some new growth before the fall, this will come at high energy cost for the tree, right? That will suck a lot of carbohydrate reserve from the trees. And also early defoliation will basically reduce the amount of fruiting spurs, fruiting buds for the following growing season. Okay, so early defoliation again come at a high cost for an orchard, and we want to avoid that absolutely. And now you see these pictures illustrating these leaves that are dropping. You see that some of these leaves have this sort of purplish or sort of dark red discoloration. These are the older lesions of red leaf blotch, and this is where the fungus is going to overwinter. This is where the fungal pathogen polystigma is going to reside. So all this leaf litter on the ground, it is basically going to serve as the next growing season sources of inoculum for the fungal pathogen polystigma. So saying that, here is a close-up on the leaf. All these leaves will show sort of different type of some fruiting structures containing spore depending on the time of the year. During the summer, these leaves contains lots of what we call pycnidia, and the pycnidia is basically, again, a sort of a fungal fruiting body. These are filled with conidia. These are non-infectious, so they don't play any important role. They probably, they're not infectious. They don't play any role in the disease cycle, but they actually may play a role in sort of the reproduction of the fungal pathogen. The real important propagule for polystigma are these ascospores illustrated right here. These are actually contained within perithecia on those leaves within the lesions. They basically sort of mature up around this time of the year, January, February, and these are the propagule that are going to contaminate new leaves in the spring, right? So these are really the important spore type for disease spread and disease contamination. And we've sort of assessed how much spores may be present within the leaves, and we're talking about millions of spores just present within a leaf. So it seems like really polystigma has a great capacity to produce a lot of inoculum and spread quickly in regions like California. So again, polystigma also is an obligate biotrophic fungal pathogen, meaning it is dependent on living plant tissue for growth and reproduction. It's not something that we can grow in the lab on culture medium. So, all our experimentations and essay will have to rely on sort of a field experiment to understand better the biology. Again, it's not a fungus that can be easily grown within a lab. So the disease cycle of red leaf blotch, as I mentioned, is sort of very straightforward. It's a monocyclic disease, meaning that there's only one primary infection cycle in the spring, but the disease cycle may start right here with those pathogen fungal fruiting structure present on the leaf litter. Those spores or ascospore within the perithecia, within the fruiting body, will mature around January. That's sort of the first catch, January, February, sort of the first catch of the spores. But again, yes, they are mature again around this time of the year, with all the ascospore being produced massively within these fruiting structures. And it's only in the spring, early, let's call it early spring, late winter, when those young leaflet emerge. These are going to be the susceptible tissue for infection. Okay, so we're around post bloom, petal fall, young leaflet emerge, and these leaves are going to be susceptible for about maybe a couple month periods, but we'll get into more detail. But the young leaves in your orchards are going to be the ones susceptible to infection, and that coincides again with the ascospores inoculum being sort of dispersed in the air. And now what's very unique with red leaf blotch is that symptom only appears something like 40, 35, 40, 45 days after the original infection, right? So a grower may be tempted to wait for symptom development to take any sort of action for disease management. But once you see symptom developing, infection has already been going on for weeks, right? Okay, so remember that once you've seen symptom developing in May or June, infection is already at least 30, 40 to 45 days old already. Then in the summer, I've mentioned these sort of pycnidia structures are forming. They don't play any role in the disease cycle, mostly a role in sort of the pathogen reproduction. And again, infectious leaf that you see throughout the summer will fall again, leading to more inoculum on the orchard floor. So for red leaf blotch in California, again, we're talking about a recent introduction that has reached within just a couple years an epidemic sort of level. We're talking about a disease outbreak. Remember, first detection here, May 29, 2024, at the sort of the border between Merced and Madera County. That's where I received a call at the time from Cameron Zuber, the UC farm advisor from Merced County. In 2024, we looked around several orchards where detected, found positive with polystigma, and we were talking in 2024 about San Joaquin, Stanislaus, Merced, Madera, and sort of the northern portion of Fresno County. But by 2025, so during last growing season, red leaf blotch was pretty much detected in all all the California almond producing counties. A few findings yet for Butte or this northern sort of region, Tehama and Yuba. Also a few detections from Kern County, but really the pathogen nevertheless had reached this sort of statewide distribution within no time. So we're understanding right here that red leaf blotch seems quite aggressive, and what we're seeing also is pretty much every cultivars of almond that we've looked at seem highly susceptible to red leaf blotch. And here I have a list of some of the cultivars from which we have reported red leaf blotch, and these include Aldrich, Butte, Carmel, Fritz, Independence, Monterey, Nonpareil, Padre, Shasta, and Wood Colony. So again, really a wide distribution for red leaf blotch within no time. And I think at this point, pretty much every almond farm's got to be sort of, ready addressing, the management of red leaf blotch in the orchards, So here I like to remind some sort of basic principle of how this disease sort of outbreak occur. But again, it looks that for California almonds, we have a very susceptible cultivar to polystigma, to red leaf blotch. It looks like the environment also of California is very conducive to the disease. And so now that we have the pathogen being introduced, again, we have all these three factors sort of reunited here to allow sort of the perfect condition for a disease outbreak. Now we, question how did red leaf blotch got here? We went from nothing around 2022, 2023 to a statewide disease outbreak. There are many reasons for disease to emerge. Sometimes it can be on some sort of specific yearly sort of weather. We've seen with some sort of aerial Phytophthora that those years, a little bit like this year, where we have this sort of atmospheric river and a lot of rain, we see emergence of sort of quiescent latent pathogen like Phytophthora that will outbreak into some specific years. But for red leaf blotch, really the pathogen must have sort of been brought into California. We're talking about a pathogen introduction, most likely through the movement of plant material, or it is possible that, somebody must have walked into the state with infected almond leaves in their pocket or something. But for red leaf blotch, it's likely that we're talking about a pathogen introduction, most likely with the movement of plant material. This is emergence also for, again, California has sort of this perfect condition for widespread disease outbreak. We're talking about, sort of a large acreage of almond, which is sort of contiguous and continuous throughout the states. So with this high density planting system, this sort of monoculture almost, where we have a lot of similar plant genetic material, all the conditions again are right for sort of a fast spread of this disease across the state. And again, that's what we are noticing within just a couple years. Red leaf blotch has gone statewide. Some of the research we have conducted last year. So remember, red leaf blotch started in 2024. And so during last year growing season, my lab between Davis and Kearney developed research with support from the Almond Board to address this new issue faced by the industry. And so the first thing that we did was to sort of, develop or provide or confirm tool for pathogen detection. That was, I would say, very straightforward. There was work published in Spain from Jordi Luque lab, which pretty much is some sort of basic PCR test with using specific primers for polystigma pathogen. And so we validated that tool. And so we had, that was the sort of the first requirement. Jumping on that research was to confirm that polystigma was actually present in the state. And so for that, we also work with CDFA and USDA for pathogen confirmation. And of course, the polystigma and red leaf blotch together were confirmed from California and using that PCR assay that was also validated. Some of the research we addressed sort of a thinking forward into disease management strategy, what we wanted to understand was the seasonal sporulation dispersal dynamic for red leaf blotch and polystigma. When are the spores of polystigma that serve as inoculum are most present within an orchard, okay? So for this, we use a very simple sort of a technique. We use microscope slides. These are covered with Vaseline, and we place 10 in an orchard with a history of red leaf blotch. And these slides are collected more or less weekly to be taken in the lab and take the opportunity to read those slides under a microscope and count the number of spores of polystigma being caught per that week during the growing season. And so then we can overlap these informations, which basically spore count, together with environmental data, so we sort of understand a little better what are the triggers for sporulation of red leaf blotch and at what time of the year, okay? And so we also now are working with the Almond board to test additional sort of new upcoming technologies. These are two spore trap type. One is by Scanit Technology here in the Bay Area. The other one is from BioScout, a company from Australia. And so these are a new spore trap technology that are equipped with sort of an inserted microscope tools that have the capacity to read a tape that is mounted within those spore traps. And so the idea is to validate this advanced technology with sort of the idea that eventually, some years down the road, we could use this type of device as IPM tool that will be basically decision support tools, allowing growers to monitor live how much spores are present in the air within the orchards, and basically receiving this sort of alert on the phone, really monitoring the fungal pathogen. This could be anything from red leaf blotch to Alternaria pathogens, and sort of allowing growers to sort of visualize live how much disease pressure is present in the orchard and help them sort of making decision when it comes to spray applications. So back to our old, more classic technology using the microscope spore traps. What we've seen in California here is a sort of almost a full growing season where we're starting in January, end of January, moving into February, March season, April, May, all the way into harvest. What you see here in blue are the precipitation, the rain mostly that occurred last year in 2025. And what you see here, the black line, are the amount of spores that were caught for that specific week for about a 1 square inch area, right? We like the 1 square inch area because this is about the surface area of a leaf, right? So what that graph shows us is when did it sporulate and how much for that specific growing season, right? So the first thing that we can observe is it seems like the onset of sporulation. This is again Stockton, California. The first big peak of sporulation were detected around the first week of March, and those overlap also with the first rain. So it seems obviously that rain is required for the sporulation of polystigma to be triggered within an orchard. And as you see again, as we're moving into April, the rain of April and early May, when it gets warmer, it seems like that's where we see also sort of the highest amount of spore catch. So this will be important when we think later disease management, and I'm going to provide more detail for disease control. But these periods where we have high amount of spores in the air will be the time where we need to think and consider fungicide application and intervention. Okay. So this is just another block a little further south. We're now in Merced County. You see again the first rain here in blue, and that's when we start to catch the first spore around the sort of the latest week of February. And then with really higher large amount of spore being captured all the way into May here, but large amount really produced in the last week of April. And so what we see here again, when we look at the number of spore we're catching for that specific week, we are about at 220 spores per square inch. What does that mean? It's within that orchard, any single leaf had the capacity to receive up to 220 spores, right? That's just to say that how high the disease pressure can be for red leaf blotch within some orchards, okay? What we see here, it's interesting. You see that there are no rain for that period of May 15, May 28, but we see still some spore present within the orchard. What we understood here is that specific block was on micro sprinkler irrigation, and those micro sprinkler were wetting the leaves that were sitting under those trees, and basically those micro sprinklers acted just like rain, and allowed also for spores to be discharged later in that growing season. So keep in mind that if you're using micro sprinklers, this also could contribute to spore release within an orchard. So again, what I'm highlighting here is, anywhere you see the speak of sporulation, this will be the time to consider fungicide applications. So now moving into fungicide control, chemical control, or any biocontrol strategy for any fungicide application, we have developed, Jim Adoskavich has developed this table that sort of allow us to better understand the optimal timing for fungicide intervention within the various diseases that are faced within the almonds. And so for red leaf blotch, the sort of three most important timing for disease management will be petal fall. What we call petal fall is that stage later, sort of at the end of bloom, at the end of full bloom, when young leaflets have emerged. We're now talking about having leaflets that might be half an inch or 1 inch in length. This is sort of the timing we mean by petal fall. So that timing is going to be absolutely critical for fungicide intervention. We're probably talking a week or 2 weeks from now at most. So really critical stage, which is petal fall. There is that other common stage that is 2 to 3 weeks post petal fall, will be also absolutely critical for fungicide intervention, and eventually 5 weeks if rain persists, right? We're now 5 weeks, we're at the end of April. If rain persists, so say that timing will be critical. But nevertheless, there's two timing that we like to emphasize that are really, really important for the disease management. Petal fall, again, when you have leaves emerging, and that period 2 to 3 week post petal fall. I get the questions also. Yeah, it's true. We realize like certain cultivar like Shasta, Independence, Bennett Hickman, they have their leaf coming in the same time as flower, right? So really the goal here is to protect those leaves. There's some of these sporulation dynamic are showing that this time of the year is not yet the sort of the highest peak of sporulation, but nevertheless, what we're trying to do with those three sprays to protect the young leaves, again, as your full bloom targeting only the flowers will have no efficacy at protecting your almond trees against red leaf blotch. So the approach here has to be an integrated disease management approach. Remember that if you have had strong history in your orchard of shottle, scab, rust, if you're trying to go after any of these other fungal disease of almond, well, if you use any of this frag group 3 and 7, 3-11, 7-11, 7 and 12, and any of the frag 3 triazole, if you use those to target shottle, scab, rust, or eventually anthracnose, this also will be highly efficacious again, red leaf blotch, if you use them as this specific timing, right? So basically what I'm trying to say here is the management timing for shottle, scab, and rust do overlap with red leaf blotch. So something to consider when you're doing your spray application, and that's the kind of question right now that we may have. We're getting a lot of rain around full bloom. There's going to be a higher pressure for this other disease, and you may consider again having an integrated disease management approach between red leaf blotch and the other fungal disease of almond. So moving into disease control, we conducted several large field trial last year using backpack sprayers or even spray bottle when we had a high disease pressure, but we conducted just about 5 trials in the field to address disease management, looking at various compounds, various active ingredients. And so for that, we worked with ag chemical company as well as biocontrol companies. We work with Bayer looking at different products. I'm not going to describe them all. I'll focus on the one that has shown great work, but we work with Bayer, BASF, Corteva, Syngenta, FMC, Heritage Crop Science, UPL, Valent, Go1, and Sanagro, looking not only at some sort of conventional fungicide, but we also looked at biocontrol solution and biocontrol agent to address this disease control for red leaf blotch. And so again, last year, remember that slide where I showed a 3-peak of sporulation. I would say last year was sort of a perfect season for us to sort of develop this fungicide trial. Remember the 3-peak of sporulation associated with rain. We basically came in this orchard with our backpack sprayer just before the peak of sporulation, just before the rain. So that's sort of a good intuition right there, but which is also should be the general sort of strategy. You look at the forecast, you see a rain coming up early in March, you're going to want to time your fungicide just before the rain, right? So that's what we did in this fungicide trial. We had multiple locations, Stockton and Merced, again, spraying the trees just before the rain, which was, again, optimal for assessment of fungicide efficacy. And so what we've seen right there, these are data for Merced. We worked with Nonpareil. On that specific block, we had 3 application timing, as I just mentioned. These were March 3 of 2025, March 19, and then we came later for the latest rain of the week of April 23. One note here, I put a star. All of this product received 3 applications. We wanted to know the product efficacy. One asterisk here, Luna Sensation only received at the time only 2 applications based on label recommendation. And so what you can see here is the disease incidence within that experimental block as well as disease severity. And so by disease incidence, for example, we mean what percent of leaves within those trees were diseased with red leaf blotch, and by mean of disease severity, what was the surface area of those leaves that was infected by red leaf blotch. What you see on top is the non-treated control. That's the water control where we have the highest disease incidence, right? 85% disease incidence in those trials. So perfect condition to test those various products, 22 product total. And so what you see in green here are the various chemicals that were tested, which show highest performance against some of the biocontrol that really didn't provide great control. So something we're going to be looking into more details, identifying biocontrol agents for disease management. But what we've seen in that first year of trial is really little to no efficacy from the biologicals. And within the chemicals, I would say we had 2 groups. We had a product with sort of a moderate efficacy, and then we had product with really absolutely great disease control. So we had about 9 products that performed greatly. 8 of these products have actually received label 2EE recommendation, label expansion. So these are now registered for this growing season. Most of them are registered, and I will sort of name them all so you remember and be prepared for that upcoming timing for management of red leaf blotch. Anything from Quadristop, Merivan, Adamant, Quash, Miravis Duo, Luna Sensation, Miravis Prime, Rhyme, and Pristine provided great disease control, almost eradicating the disease from this orchard. So we are equipped with great tools, good tools for disease management, but unfortunately no product yet sort of coming out of any of the biologicals. So something that we're going to look into more detail this upcoming year. This is a different location, also just to see sort of the consistency of this product efficacy. 3 timings also of a spray application, just 2 for Luna. Here we added new film P as a sort of a surfactant, sprayed a sticker for some of these biocontrol agents. And again, what we see is really little, but improve efficacy from some of the biocontrols down there, where we start to see some disease control. If you look at Romeo and Gleam. But overall, again, really the best top result from these trials, again, were acquired with anything from Quadris, Top, Luna Sensation, again, Quash, Rhyme, Pristine, Miravis Prime, Miravis Duo, and Adamant. Okay, so the very, very, very good products here. This is another orchard where we actually try to come in a little later. So we had a delayed one single application within the orchards. And what you can see, disease incidence remain high, within sort of all these good product using only one single application. So that's something to remind all of us that probably 2 to eventually 3 applications are going to be the minimum requirement. Is 2 are going to be the minimum requirement for disease management to stay on top of red leaf blotch, as a single application doesn't show enough disease control. We also get the question often is about how, cultural practice. How about I get rid of all these leaves on the ground, as they serve as the, sources of inoculum, with zinc grower after harvest, really cleaning up very well these orchards, not leaving any single leaves on the ground. So I think these are definitely great initiative. They can reduce the amount of inoculum that's going to be present within the orchard, and that could be some sort of physical elimination of the leaf litter. Some growers may consider zinc sulfate to hasten leaf fall and give more time for the leaf to decompose within the dormant season. In scab, for example, apple scab, people have used urea, maybe things like lime sulfur to accelerate leaf decomposition. They are a beneficial microbe that can be used as biosanitation. Nevertheless, this type of practice can really only be effective if we use them over a large scale, large, wide area, right? Basically, in other words, if you're doing lots of efforts to clean up your orchard, but your next door neighbor is not doing it, then this is going to have sort of a little effect on your capacity to get rid of the inoculum, as we've seen that red leaf blotch can spread some distance. Okay. What we recommend also for growers up north or down south, if you don't have the disease yet, and if you're moving any harvest equipment or spray equipment from Fresno, San Joaquin County, also you could also consider cleaning up this equipment, not to bring infected leaves within a clean orchard. Also, things to consider. What we are planning next, that's something we're going to start this year, is understand the duration of leaf susceptibility. And basically, the question here is, if I have leaves that emerge right now, that first week of March, if rain persists, how long will a leaf be susceptible to red leaf blotch? You can imagine of a leaf sort of developing this waxy cuticle layer with time, and so we expect that an older leaf will not be as susceptible as the young leaves. So we're going to put again another couple of field experiment to determine, when does a leaf that has emerged in March stop, for example, to be susceptible? Does it take 6 weeks, 8 weeks of a leaves, 12 weeks, all weeks not to be susceptible? That's what we want to look to sort of overlap this information with the spore trapping study to really sort of optimize again our timing of fungicide application, with the main question is hopefully being able to take these down to 2 instead of 3 sprays that are required for disease management. With this, I will conclude it right here. The main point of this talk is, red leaf blotch is a new and serious disease for California. We're talking right now about a disease outbreak. The disease is here to stay. It's not going anywhere. We're going to have to live with red leaf blotch within our almond industry at the time. And then it's sort of a little bit of in terms of disease management, new approach. We knew that a lot of growers were maybe eventually neglecting this timing of the year for disease management with, high cost of fungicide intervention, lower incomes within almonds. The grower are more looking to do sometime 2, 3 spray within a growing season, but we're gonna have to, consider again those pedal fall, 2 to 3 weeks post pedal fall, and eventually 5 to 6 weeks post pedal fall as really important now critical timing for red leaf blotch management. Think of this again as an integrated approach. Any strategy to go after rust, scab, and short hole using specific frag groups will also help with red leaf blotch, right? We're talking again about the 3 and 7 fungicide, the 311, 711, 7 and 12, and the Florent Triazole, which are the most effective. I mentioned a few products. 8 have been registered, so we're all ready again to tackle red leaf blotch for this upcoming growing season. Management is going to start soon. We're looking at a few days or weeks from now to start initiating those sprays to stay on top of red leaf blotch. Use active ingredients smartly, right? The general recommendation is not to use a single frag group for more than 2 times within a growing season. Try to avoid repeating application of the same frag group to avoid development of fungicide resistance. And again, more research is happening right now. We're only at year 2 of our research project. We already acquired quite a bit of information, but we're going to fine tune the knowledge and specifically looking at more biologicals and try to really optimize the number of applications. I'm fully just taking it down to 2 to stay on top of red leaf blotch. And then again, remember that fungicide applied during bloom or after symptoms are visible are not effective for the management of red leaf blotch. And again, during bloom, what I mean there is if you target the flowers and if leaves are not out, those bloom sprays are not going to be effective. With this, I'd like to thank everyone for attending the webinar, and I'd like to thanks UCNR and UCIPM for supporting that event, and I'd like to also thank the Almond board for supporting this research, as well as a large team of graduate student technicians, project scientists in my lab working full-time for about a growing season to address this emerging issue. A big thanks also to numerous farm advisor Cameron Zuber, Brent Holtz, and the numerous collaborators, growers, and and PCA helping out making this research possible. So with this, now I'll be glad to take any questions that are on this specific issue. Thank you.
Thank you, Florent, for excellent presentation. And there are a few questions I'll try to go through. I think some of them you've already responded, but maybe you can repeat it just so that everybody understands. First one, so this disease is from ascomycetes. Does it mean that the life cycle is similar to apple scab, which is also ascomycete with perithecia in leaf tissue, right?
Yeah, I would say there are similarity on apple scab. I don't work on apple or apple scab, but from general knowledge memories that I have on this disease, yes, we're talking about something very similar. The pathogen is an ascomycete. It will grow on the leaves, and it's only within the leaves that it will form those perithecia or fruiting structures that will produce ascospores, a form of spores that will be the main inoculum sources for new or young leaves infection. So that's really sort of the same idea, same principle as apple scab.
Thank you. The question, again, I think you talked about it a little bit. How did it happen that the pathogen spread so quickly, practically within 1 or 2 seasons throughout the whole California?
That's the great question. We're all sort of scratching our head to figure out, how come he went statewide within no time? That's really unprecedented. We never seen anything so aggressive. So I think he has to do again sort of that disease triangle. We have a very conducive environment for California. The pathogen has the capacity to spread, very fast, very quickly. A single leaf again can produce millions of spores. And again, we have this sort of continuous sort of production of almond trees within the state. So all these factors together with the high susceptibility of the almond cultivars, pretty much all the almond cultivars are highly susceptible. This could explain the spread. We know that, equipment for harvest spray sometimes is contracted, so an equipment can move from the Central Valley north south, and so these are all some of the reasons that can explain that sort of a fast spread of red leaf blotch within California.
Okay, thank you. The pathogen is an obligate biotroph. Does it have any alternate hosts known in California?
No, again, it's really highly specific, and it will only affect Prunus dulcis, meaning almond trees, not any other stone fruit, not any other Prunus species. Only it would just affect almond trees.
Thank you. Could you summarize quickly the environmental conditions that are favoring the red leaf blotch?
Yeah, so basically what it takes for red leaf blotch is rain. The effect of temperature doesn't seem to be a limiting factor. We know that for specific disease, a cold winter, a cold spring, may favor more botrytis jacket rot than it will favor monilinia. For polystigma, we definitely see more spore being produced later in the spring, like in April, right? The peaks are much higher, so there could be a general impact on sporulation, but it's not sort of a absolute limiting factor, right? If there are rain, there will be spores of polystigma in the air, and so rain is really the main trigger we want to be focused on. Temperature will have overall of a moderate effect when it comes to the disease biology.
Thank you. A clarification on the petal fall. When you say petal fall, does it mean the very end of the petal fall when there are no more blooms and the leaves are starting to grow?
Yeah, so I get that question often. So the table is what we have on the IPM website, is what we've used over the years to try to describe those various phenological stages. But by petal fall, yeah, we usually mean the end of bloom when leaflets have emerged. Okay, so I think the idea with red leaf blotch, the ideas of those first application is to protect as much as those young emerging leaves. Okay, and so I think having this sort of cutoff of having about 1 inch leaves that have emerged a good amount. It's like with full bloom. Do you want to make your full bloom when the flowers are out 20%, or do you want to spray when 80% or 100% of the flowers are out, right? So that's the same for red leaf blotch, where we are trying to target all these emerging leaves. So yeah, sort of a later and end of bloom where most of the leaves have emerged. This is your ideal timing. Remember also that the peak of sporulation comes around that first week of March. So I think, yeah, we're looking around that sort of last week of February, early week of March let's talk about nonpareil, for example, right? We see that, again, I've mentioned Independence, Shasta, Benedictman seems to push leaves in the same time they push the flowers. So you may consider maybe a little bit earlier spray. Nevertheless, a lot of the product we are using have systemic activity again. So I would rather favor sort of late petal fall than an early petal fall, right? You don't want to come too early when you only have 20% of the leaves out. Again, I would think of last week of February, early March for California overall as being the optimal timing.
Thank you. Question that came through chat. Will this information work for red blotch in vines and the same fungicides?
Red blotch in vine has nothing to do with that disease. And again, the pathogen we're talking about is a fungal pathogen specific to almond. Red blotch in vine, although the name sounds familiar, red blotch in vine is a viral disease, virus associated disease. So the biology is completely different, and none of our fungicide strategy will work for red blotch of grapevine. Completely different system.
Thank you. Could a controlled orchard be infected again in subsequent seasons?
Could a what? A manage?
Yes.
Yeah. Yeah, so those disease it's most likely impossible to get rid of these inoculum sources of red leaf blotch. Even within an orchard, if you try to break the leaves, there'll all be little pieces of leaf that will contain the fungus. The pathogen may come from your neighbor. The pathogen may come from maybe 10 miles away. If your spore will have the capacity to fly with the wind. So, you want to think of red leaf blotch management as sort of a yearly sort of consideration, especially knowing your orchard history. It's always important in IPM to understand your orchard history, how much disease you had the previous year. But at this point, red leaf blotch is so aggressive, so widespread that I think it's something that we're gonna have to consider years after year, even even if you have had a low history on the previous year. So for those orchards up north or in Kern County right now that didn't have much disease last year, I think it's maybe time to consider intervening, having some intervention for red leaf blotch management this year, as the pathogen again can spray from very quickly. So.
And the last question before we start before I share the final test, does the level of red leaf blotch severity and defoliation translate into a measurable yield loss?
So we try to work with some PCN growers to get that information specifically this year. It was difficult to assess. We had a very, very hot summer in 2024. It seems like the yield that dropped throughout California independently from red leaf blotch. All that we know so far is discussion from our colleague in Spain. Defoliation from a year to another, especially if repeated, can lead up to 30% yield loss. That's the information we get from colleague in Spain, something yet to be sort of assessed here in California. But again, defoliation usually, especially if repeated, can lead to significant yield loss.
Thank you very much, Florent, for presenting today, and thanks everybody for attending. Enjoy the rest of your day.